So hello, everyone. Super excited to be here at this month's community hours, specifically to talk about citing BDC in papers. So as Paul had mentioned, I work as part of the Picture platform on Biodata Catalyst, and that platform, it was created by Paul Aviak's lab at Harvard Medical School. So very happy to be here. Next slide, please. So let's just jump right into it. Where can you find how to cite Biodata Catalyst in a paper? So I provided a few links here on the slides, which you should be able to access after this talk, but you can always check the Biodata Catalyst Gitbook documentation. The link is provided there for the most up-to-date version of the citation. And specifically, it is under the citation and acknowledgement section of this Gitbook where you can find this information. Next slide, please. So hopping into it, how can you cite BDC? And please note that this is as of today. This could change in the future, but the current citation is listed here. And I got this directly from the Gitbook documentation. So again, that's a good source of truth to consult if you're ever curious. But you can see that you will cite the National Heart, Lung, and Blood Institute, and specifically NHLBI Biodata Catalyst. There's also an option to add an acknowledgement, a sentence of acknowledgement in the acknowledgement section of the paper. And here we've provided the wording as well for how to contribute or how to acknowledge Biodata Catalyst on your paper. And I will be going through 2 examples of these in actual publications that have been created as a result of BDC. So that way we can go through an example. Next slide, please. So I also wanted to quickly discuss what is a DOI. So if you saw on the previous slide, there was a link at the end of the citation. And I've also added the BDC DOI as of today on this slide here, which you can see is that 10.5 some numbers, Zenodo dot some numbers. And so what a DOI is, it's a digital object identifier. And DOIs essentially allow for us to uniquely identify and access things reliably. And so this is really because URLs can actually change over time, but DOIs are persistent. So this way we always make sure if we reference something in a publication and list a DOI, it will always be able to reference the thing we are linking to, regardless of if something changes. So DOIs are persistent and therefore good to use in publications. And here the BDC DOI, as you can see, links to a Zenodo page. If you're curious and click on the link that was listed in the citation, it will take you to a Zenodo page, but that basically hosts an abstract that was written about the BDC ecosystem. And so, you know, if you add this citation and this DOI as a reference in your publication, people that are curious can link and click the link and learn more about the BDC ecosystem through that abstract. Next slide, please. So here I've added a few examples of publications that cite BDC. So this first one here is actually an analysis reproduction of the ORCID clinical trial that tested the effect of hydroxychloroquine as a COVID-19 treatment. And so I've listed the DOI here if you're curious about accessing this publication. But you can see here, I've cut out all the middle stuff and went down to the acknowledgements. And so this author here added BDC as an acknowledgement using the same phrasing. So you can see here the NHLBI Biodata Catalyst ecosystem is added as an acknowledgement in the manuscript. Oops, excuse me. Next slide. And so on the other hand, you can also add BDC as a citation or a reference in your list of references. So that's what this author here did. So this is a publication about the survival of sickle cell disease patients after hematopoietic cell transplant using the HCT for SCD dataset, which is one of the sickle cell disease related datasets that we have available on Biodata Catalyst. Again, I've referenced the DOI here if you're interested in reading more, but you can see here that skipping down to the references section of this paper, if you add a if you add the citation that was listed on the Gitbook documentation, it will show up as something like this. So you can see that there's the NHLBI Biodata Catalyst and Zenodo information. And so they can click, or readers that are reading this manuscript will be able to click to learn more about that citation and learn more about Biodata Catalyst. All right, and then as far as the next slide, I think that concludes my section. So yes, so that's all we've got on citing papers. And if you have any questions, feel free to throw them in the chat. I can answer them. But I will hand it over to Dave to discuss Seven Bridges projects.
Thank you, Emily. It was wonderful to learn about DOIs. It's relatively a new concept for me, so it's great to learn how we can cite BDC in our publications. So my name is Dave Robertson. I work for a company, Valsera, and I'm the community engagement manager for Biodata Catalyst powered by Seven Bridges. So what I want to talk to you today about is how you could use the Seven Bridges platform and to share projects and apps, and why you might want to do that. So I always like to start out with like a motivating problem statement. So as everyone knows, analytic replication of science is pretty difficult. This is putting aside all the wet lab stuff, right? So even if you're starting from the same data, sometimes reproducing the results is still difficult. And why is that? It's because a lot of times researchers are forced to reinvent the wheel. There's a lack of access to methods, analytical details, raw data, and other research materials. In addition, not everyone knows the details of the specific models or the software that was used. So that's sort of a bit of a learning curve, and it makes replication difficult, but also makes using a similar approach in another study very hard as well. So I'm just quoting from a Nature paper here. I put the link for, and you can get the slides in the forum later. But the potential solution is to have robust sharing of data, materials, and software, as well as other tools. So, we'd like to have, since there's no demonstrations today, so we're going to have some time at the end for discussion. So I just want you to think about, have you ever experienced difficulty replicating analysis methods? And I'm sure lots of people have. It's a really difficult thing sometimes. So just keep that in mind. So one way that Seven Bridges could help you here is that you could use projects to share your analysis methods, your data, and your workflows. So on Seven Bridges, project is sort of like the main organizational unit. You can think of it as a container for your data files, your apps, which is another word for scientific software, and your analysis tasks. So a task is simply an analysis that's already been completed, and you want to save that and share it with other people. So an immediate thing you could do is simply invite collaborators to a project, and there's no limit to the number of members in your project. So if you're inviting people, just be careful if you don't want them affecting your project, just give them sort of read only or read and copy permissions. This means that they could sort of copy the data out. Of course, make sure that it's open data, and I'll talk about that a little bit more. But it's a great way to share your results. But what if you wanted to share sort of more publicly? As a BDC user, you have the opportunity to broadly share your analysis examples, notebooks, workflows, and data through a feature called the public projects. And now we're working on a better interface for here, but currently it's a drop-down menu. And here's some of the public projects that we have. They range from everything from introduction to SAS, which I need to read through because I'm not very good at SAS, but there's a nice introduction to that created by the SAS group. There's a lot of machine learning examples. If you want to see how the TopMed Freeze 8 variant calling was done, There's a project for that. There's a picture API, which gives you notebooks. So this Emily and the picture team created this and published it on 7bridges. So this actually has the Python notebooks that and examples of how to interact with the picture feature inside one of your 7bridges projects. There's a COVID-19 image segmentation and deep learning project, which I've demoed a bunch of times. This is created by Matt Satuski. That's a very great way to learn about image segmentation. And then, of course, the Genesis tutorial, which is one of the most popular public projects we have. So this is kind of like a call to action also, because I think we need more of these public projects. And if you're working on something and you'd like to share it with the community, you can do that by following an example here. So you would prep your project a certain way. And if you want more details here, there's a reference down here at the bottom right. But things to think about. So give your project a title. Just, don't say it's Dave's project or Dave's workspace. Give it a sort of short but informative title. Use the description. So the description has the ability to have bold texts and different sections and links. So really provide some good documentation there. Now your files. So you can have any files that you want to share in your project. Just make sure that they're ideally share only open access files, but these could be open access files that are hosted by NHLBI. So we have a few different open access data sets, including 1000 genomes. So you can share a lot of 1000 genomes data in your public project if you're using that as an example data set. Of course, you can share your apps. So these could be apps that you have customized or just apps that are already available on the public apps gallery. But you can also share Data Studio. So Data Studio is a way to run SAS, RStudio, and JupyterLab. So you can share those environments, which includes scripts and sort of worked out examples as well. So consider all those things when you're ready to prepare your project. And then when you're ready to publish your project, the current method is to send support at 7bridges.com an email with a link to your project. And what's going to happen here is that 7bridges behind the scenes is going to make a copy of your project, and they're going to publish it to that public projects dropdown and make it accessible to any user with a login. Now, if you need to update, you have to email support again. The benefit here is that sort of SB will control any hosting or any associated costs. There's no cost for the NHLBI hosted data, but there may be some derived data. So SB will sort of take care of those storage costs. Updates are not immediate though. The 7bridges will need to make another copy whenever you want to update this public project. It's something just to be aware of. That said, you can also publish common workflow language apps. So this could either be a single tool or an entire pipeline of multiple tools. And this actually gives you really fine-grained control over what you want to have published and also how many updates and revisions you want to do. So first let me say that here's a sort of like a screenshot of part of the public apps gallery, 2 apps that I just saw. And if you publish your own apps, you'll have a published by whatever your institution is. So it's up to you. It could be your PI's lab or whatever the name of your institution is. So here's 2 examples of published by UWGAC, or the University of Washington Genome Analysis Center. They're by far the most prolific publishers for the 7bridges Public Apps Gallery. I think they have about 20 apps or so. But here's 2 apps, allele frequency and heterozygosity by sample. So how this works is they have one what's called a commit project, which only they are members of, and all the apps in this commit project will become accessible in the public apps gallery. And you can try this out now. The great thing about the public apps gallery in this example is that there's no login. I made a quick link here. If you go to bit.ly/bdc-apps, that'll take you to a longer URL, which is a completely open URL, and you don't need to log in. So the user maintains control over the revisions and also control over the URL. So this would be, going back to Emily's example, this would be great for creating a DOI. I sort of glossed over a lot of details here, but feel free to email me at david.robertson@bellsera.com for a sort of a more detailed walkthrough. And some of those details, you don't have to do this, but what University of Washington did was they connected their GitHub repository and 7bridges. And the way they did this is using a piece of software called 7bridges pack, or SB pack. And you run this command and it will actually pull. So on the left side here, there's their GitHub repository. So basically every CWL app, and this goes for other workflow languages, every CWL app is a file. So you can see some of these allele frequency, heterozygosity by sample, missing rate by sample. You can run the SB pack tool and it will push it to your commit project, and then immediately it's available in the public apps gallery. So looks like they don't have any revisions here, but you can have multiple revisions and that'll all be saved for the end user, as well as if you may not be able to see this, but it actually says what commit what the commit message was for that particular commit. So I think that is it. Let us know after Samantha's presentation if you have any specific questions. So I want to pass it to Samantha from the Broad Institute.
Thank you. Hi, everyone. My name is Samantha Velasquez. I work at the Broad Institute, and I'm a product support specialist for Terra, which is our biomedical research platform. Next slide, please. So first, what is a workspace? For those not familiar with it, it's basically your computational sandbox with everything you need to complete your project. So you can have data analysis tools and documentation. You can link to data in the cloud, keep data organized with spreadsheet-like tables. You can visualize and analyze your data in interactive analysis tools like Jupyter Notebooks, RStudio, or Galaxy. And you can find and run bulk analysis tools with Whittle workflows. And you can also share your workspace with a user or group of users, which will allow all of you to actively work together in the same workspace. Next slide, please. If you want to share your workspace with a broader audience, you can share your workspace, or you can make your workspace public, which gives all registered Terra users read-only access to the workspace. The workspace will also be listed under the public tab in your workspaces page on Terra. And to make your workspaces public, you can just contact support@terra.bio. Next slide, please. If you have a more curated workspace, you can feature it on Terra, which makes the workspace public and also adds it to our featured workspaces showcase page. So there are 3 types of featured workspaces on Terra. First is analysis focused. These are generally associated with pre or post publication studies to highlight your work and allow users to reproduce it. One example of this on Terra is our COVID-19 featured workspace. Another type of workspace is data focused. These introduce users to specific open or controlled access datasets available in Terra. So some of these workspaces include Target or TCGA. And lastly, we have workflow-focused workspaces, which showcase certain Whittle workflows or Jupyter notebooks with sample test data that users can run on their own. Some of these include JTK, best practice pipelines. And how these differ from public workspaces, like they mentioned, they're listed in our featured workspaces showcase page. Users don't have to be logged in to see the featured workspace dashboard, which means they have all the documentation they need to see what's in the workspace, what data is there, what analysis is there. And there's also a longer and stricter review process, which includes a review by Terra support and UX. Next slide, please. So this slide just outlines the process for getting your workspace featured. There's a template you need to follow and a form that you'll need to fill out once your workspace is ready to be featured. All this information is available on our support site and also linked on this slide. And I think that's all I had.
Great. Thank you so much to Emily, Dave, and Samantha for those great presentations. We'd now like to open up the floor to all of you for any questions or further discussion on these topics. Some of the potential questions that were raised earlier: have you ever used a public workspace or project? Which public workspaces do you wish were available? And have you experienced difficulty replicating analysis methods? If there's any questions or topics, we'd be happy to discuss. Yes, Brandon, please go ahead.
Hey, thank you. I had a quick question, and I might have missed this in your presentation, but if I have an app that I built out that I modified but didn't build from scratch, how does that get published? Would I upload the code to a GitHub, or would I just directly publish the app from that?
It's a great question. We probably need some better documentation for this. So GitHub is not a requirement. What you would do is you would make a project that you own, or maybe a project. Maybe if you're collaborating with other people at your institution, maybe you make one, and you sort of become the point person for Biodata Catalyst apps at your institution. But this will be marked as a commit project. So it's not a project that you would do your work in, but sort of like a repository of your apps. And any change, like all apps in that project, will immediately be made public. And it's one support. It's one email to support basically will make this happen, and they'll probably come back to you and ask just a couple simple questions. One is, you're going to get a what's called a pill. I think it's a little icon that says, if you're from UNC, it could say UNC, for example, or it could say published by, whichever lab you're a part of. And then from then on, anything that's public in that project will automatically be published. So that's why they suggest, or why we suggest, having sort of like a scratch project where you're testing things, and then a commit project, and then all your analysis projects would be completely separate.
Okay, that makes sense. Thank you.
No problem. Any other questions? Yes, we will definitely be posting. This is a direct message to me, but Paul's going to make sure like everything gets posted to the forum, and I'll send you an email directly.
Yes, and once again, thank you, Dave. Once again, I'll put in the chat here links for the community forum, along with links to join our ecosystem and to contact the help desk. Again, joining the ecosystem will help keep you informed about future community hours like these, along with getting the newsletter and other really vital ecosystem news. So are there any additional questions or discussion topics today? Okay, well, it sounds like we can wrap up a little bit early and give folks some time back. We thank you all for joining and hope you found today's presentations informative and helpful. Again, please feel free to reach out to the help desk with any additional questions. Thank you all and have a great day.